the Early Care Research Fellow from Cancer Institute New South Wales. He works here at the ImageX Institute and is doing some research in rotating patient geometry and also on the MRI LINAC. So as always, if you want an attendance certificate, Julia will put her email in the chat, and then please send her an email and she will respond with your attendance certificate. If you have any questions, please put them in the chat, and then I will enter a poll at a time that's convenient, or you can wait until the end, and then there'll be a Q&A session at the end where you can ask your questions to poll yourself. Feel free to either leave your camera on or on or off, and please make sure that you're muted, but I think everybody is muted already. So without further ado, Paul, I'll let you take the lead.
Thanks, Yusuf. So welcome everyone. Normally I give this radiobiology lecture as the first one in the series, but this time it's the last one in the series. Normally I assume all the students know absolutely nothing about radiotherapy, but this time you all know everything already, so we'll see how we go. Probably have no idea what radiobiology is, which is fair enough. It's something that I've never heard of as well when I first started in this field, but it's actually a very, very, very interesting field, pretty unique in all of science, I think. Okay, so what is radiobiology? If we go on to trusty Wikipedia, it says it's the study of ionizing radiation in living things, especially the health effects of radiation. So that's pretty apt model description. What radiobiology really aims to do is to look at how cells, organs, and organisms respond when they react to radiation, and just as importantly, to try to model these mathematically. So it's a really kind of interesting topic that's kind of intersects math, biology, and physics. So it's kind of unique, and there's not that many people in the world that do radiobiology because, yeah, it's hard to find people that specialize kind of in all three, and you really need like in-depth knowledge in all three in order to kind of be someone who studies radiobiology. So there's kind of a couple of important things to note about radiobiology. First thing is that it's really kind of the fundamental underpinning of radiation therapy treatment for cancer. So if you think of radiotherapy as like building a house, this is really like the floor plans that you really base the whole thing around. So making an advancement in radiobiology is very difficult because you need knowledge in all these three fields. But if you manage to make some sort of advancement, because it's a fundamental building block, it's going to affect the whole treatment chain all the way. So kind of the concept of radiobiology is this idea that physical dose is not equal to biological dose. So what this means is that when you make a measurement for the amount of radiation, what you're really measuring is physical dose. So when you hear people saying this is a certain number of gray of radiation, what they're referring to is the physical dose. But on top of that, there's something called the biological dose. And the biological dose takes into account how cells react in an organism. So that includes what type of cell it is, where the cell is in the body, what that cell is doing, what that environment is. And depending on those factors, the biological dose can be wildly different even for the same amount of physical dose. So physical dose, you can see in this image, how you would measure it is something simple like a water tank. When you talk about radiation in terms of physical dose, it's just depositing a certain amount of energy in a certain volume. So 1 gray of radiation, by definition, is when you deposit 1 joule of energy in a kilo of water. And that's the same no matter where you are in the world, no matter if you're working on CERN or if you have a uranium source. 1 gray from either of those is always going to be equal to 1 gray. Whereas, as you'll see in this lecture, the biological dose, depending on a number of factors, can change greatly. Okay, so radiobiology, as you might have gathered from that, is a whole kind of unique area of research, and it's not something that I can explain perfectly in 1 hour, but hopefully what we can do is cover the basics, and by the end of the lecture, you'll be able to at least answer these kind of 2 questions which are fundamental to radiotherapy. So if we see radiotherapy, the overall goal is to cure cancer by delivering a lethal dose to the tumor and minimizing dose to healthy tissue. So that's the aim of radiotherapy. And what radiobiology will help us explain is, 1, how does radiation damage and kill cells? So what are the actual mechanisms that radiation can kill cells and kill tumors? And then number 2 is mathematically how you can model this process with the end goal to improve radiotherapy treatments. So by modeling part 1 mathematically, you can actually improve radiotherapy treatment overall. Okay, so before we get there, let's just explain what radiation is very clearly. So radiation is simply the transfer of energy through a medium. But when we talk about radiotherapy, when we talk about radiation, it's in the context of radiotherapy, which really means in this electromagnetic spectrum that covers all types of radiation, we're only really talking about ionizing radiation, which is this purple part that begins just after ultraviolet. So starting from X-rays and on to gamma rays. So anything below this we call non-ionizing radiation. And from a radiotherapy point of view, that means it's not going to interact with the human body. So it's not of particular interest or concern. But above this sort of purple section, that's when photons from this radiation is going to start interacting with the human body. And most importantly, it's going to undergo ionization. Okay, so ionization is a process of scattering. Scattering is sort of how radiation interacts with the human body. And there's a number of different scattering factors, and that depends on what type of radiation it is. Like, for example, we have X-rays usually in radiotherapy, and then also what kind of medium that radiation is interacting with. So in the case of the human body, that's mostly going to be water or carbon. And with that combination, the most prevalent type of interaction is going to be Compton scattering. So Compton scattering, you can see, is interaction that happens when the energy of the radiation is high enough. And this incident photon will come in here, interact with these electrons. And what will happen is that with sufficient energy, it's going to kick some of these electrons out of their shells. And then this incident photon will become scattered, and an electron is going to be ejected from the atom. So this is the process of ionization, and this is how the human body is going to be damaged, because if you follow this scattered electron, what will happen is that it's going to interact with DNA. So this electron that's been ejected from the atom is going to have a lot of energy imparted on it, and a number of things could happen. It could interact with a DNA. One of those is that it could interact with a DNA strand. So if this was to happen, these charged particles these electrons can make free radicals, and that bonds with oxygen to create DNA strand breaks. And this is what ultimately is going to kill a cell, because when these DNA strand breaks, one of the outcomes is that it will cause the cell to die. Okay, so if you think about it in your body, there's not really in terms of mass, there's not really much DNA. So the chance of this actually happening is not that high. The chance for an electron to interact with a DNA strand and cause a break is not that high. But what we have a lot of in our body is water. So really the majority of interactions are going to be electrons interacting with water. So these electrons can cause a secondary ionization reaction that can cause this hydrogen atom and this hydroxide molecule to split off. And this hydroxide molecule can also make free radicals and cause DNA strand breaks. So this second interaction, which is called indirect ionization, is much more common than this direct method. But the end result is the same. The end result is that when ionizing radiation enters the body, it's going to undergo scattering, usually with the Compton effect. That's going to eject electrons, and the electrons, either through direct methods or indirect methods, is going to cause DNA strand breaks. Okay, so what happens next? So DNA strand breaks, when they occur, can either occur as single strand breaks or double strand breaks. So single strand breaks are when one side of the DNA helix is broken, and this is usually non-lethal. So if you know the way that DNA repairs, it can easily sort of match bases correctly and then repair the DNA strand break over time. And this means that the cell will eventually probably survive. The other type of strand break is the double strand break. So this is when either a single interaction causes two strand breaks, or if a strand break occurs and then at a later point in time, another strand break occurs. When this happens, sort of the DNA can basically be chopped off, and it will still try to repair itself if there's another copy. But this can sort of lead to misrepair. So if you imagine kind of the double strands flipping when they try to repair themselves. And this type of strand break is basically lethal damage. So repair can still occur, but it's much more difficult. And often the cell will sort of know this will happen and basically commit suicide. It's called cell apoptosis, and kill itself to sort of prevent mutations from happening. Okay, so this is very important. Okay, so we're just going to stop here for a second and think about what I said. So it might sound kind of simple that depending on what the cell is doing, it can either repair this double strand break or it can't repair this double strand break. But this is actually a very good indication of what I was saying before about how physical dose and biological dose are different things. So if you think about this cell cycle here, this is what cells do when they replicate. So this G in the inner circle here, this blue G stands for growth, where the cell is growing. S is synthesis, where DNA is kind of replicating. And then there's a second growth phase. And then this M phase is mitosis, where the cell splits. So what happens when there are double strand breaks is that whether the cell is able to repair the double strand breaks depends really on which part of this cell cycle they're in. So in some phases of the cell cycle, there is going to be a spare copy of the DNA where double strand breaks can be repaired. And in other parts of the cell cycle, there aren't going to be a copy of the DNA, which is more likely to result in cell death. So if you look at this chart, depending on what phase the cell is in, even for the same amount of radiation, the number of cells that would survive is wildly different. So just as an example, if a cell is at this mitosis phase and I irradiate the cell with a certain amount of radiation, 40% of the cells will survive. And that's because there is a copy of that DNA where double strand breaks can be repaired. If I irradiate the same cells with the same amount of radiation at the start of this synthesis phase, for example, only say 10% of the cells are going to survive. Yeah, so this is an example of how biological dose is different to physical dose. So I've delivered the same dose, but I've gotten a different effect. And we're not kind of talking about, it's 10% better or a few extra cells survive. This is sort of, this is sort of a cartoon graph, but here, kind of 4 times the amount of cells survives. And you'll see later in graphs, you're going to have sort of factors of 10 or factors of 100 difference in survival, depending on various factors. Okay, so radiobiology is kind of about graphing and modeling. So I'm going to introduce a graph here called the surviving fraction type of graph. And this is very common in radiotherapy. So how this works is on the y-axis is surviving fraction, which means if I have a certain number of cells, what percentage of them will survive? And on the bottom here is a certain amount of dose. So how you would read this is say, if I deliver 6 gray of dose, on this purple line here, which represents late second stage growth, about 1% of the cells will survive. Whereas if I give 6 gray and we're in this yellow asynchronous mixture stage, then something like 50% survive. So that's cell cycle. Okay, so yeah, so this is a concept that's called relative biological effectiveness or biological effective dose. So what relative biological effectiveness is basically giving the same dose, so giving the same sort of x-axis amount and looking at how much damage you're causing. So taking the same x value and seeing what the y values you get in this graph. The other way to look at it is biologically effective dose, which is sort of the other way around. You say you want to achieve a certain surviving fraction, so say 10%, and you look at what amount of dose is required to get to that survival rate. So that's, so you would say, if I want 10% survival, the biological effective dose is 4 gray in this purple mitotic stage, and it's something like 11 gray in this yellow asynchronous mixture stage. So these are sort of in radiobiology, different ways of describing biological dose. Whereas if you were looking at physical dose, you just kind of look at 1 gray, which is always 1 gray, no matter where you are in the world. So another kind of important factor, which I'll also explain later, is oxygenation. So if you remember when I was talking about the strand breaks happen when electrons interact with oxygen. And without oxygen, even if you deposit huge amounts of radiation, it's just not going to cause cell breaks. The DNA breaks, and it's not going to kill the cells. So, for example, aerobic cells, which have a lot of oxygen, you can give something like 10 gray, and only 1% of the cells will survive. That's this solid line here. Hypoxic cells, which are these open circles, are cells that are starved of oxygen. So if I were to give 10 gray to hypoxic cells, something like 80% would survive. So again, when we're talking about biological dose, you can get huge differences depending on what environment these cells are. Another kind of factor that's very common in radiobiology is this idea of linear energy transfer. So linear energy transfer describes how penetrative radiation is. So if you know radiation, there's lots of different types. So we're very familiar with X-rays, but there's also beta particles, which are electrons, And then there's alpha particles, which don't penetrate very far. So linear energy transfer is how many ionization events occur per unit of distance when radiation enters a medium. So for example, gamma rays, when you get photons entering the body, for example, the average distance for a gamma ray collision is 164 millimeters, whereas with an alpha particle, it's only 0.04 millimeters. Okay, so in this picture, it kind of shows the difference between types of radiation which have low linear energy transfer and types of radiation with high linear energy transfer. So if you look at these circles, again, this is just a cartoon, but if this circle represents a part of the body that have equal volume, so equal areas of equal size, a bunch of low energy transfer radiation will deliver their radiation, their ionization effects in this sort of spread out area. Whereas a high linear energy transfer type particle will have all its interactions very close to its path. So in this case, you can see this volume of the body, we have the same number of interactions. So in terms of physical dose, we've delivered the same amount of radiation to both volumes. However, if you look at the biology of this high LET situation is going to cause a lot more damage. And that's because these interactions are occurring close together. So if you think of DNA strand breaks, there's going to be a lot of double strand breaks potentially in this area here near the high LET. Whereas in the low LET, the number of single strand breaks might be similar, but the number of double strand breaks, because it's kind of spread out over this volume, is going to be a lot lower. So again, same physical dose delivered to both these circles, but you're likely to get a different biological result in these two situations. And following on from same vein, you have the surviving fraction curve. So as you increase your linear energy transfer, which means the radiation becomes less penetrative, but deposits its energy immediately, you're going to get a lot more cell death for the same amount of radiation. So, for example, if you deliver 4 gray, well, it says red here, but we'll say it's gray. You're going to get, with X-rays, you'll get something like 60% survival, but with alpha rays, you're looking at something like 0.1%. Okay, so that's sort of the end of part 1. So hopefully, after listening to that, you can answer the first question in radiobiology, which is how does radiation damage and kill cells? So damage is really caused by ionizing radiation. So that's radiation that has more energy than ultraviolet, so X-rays and gamma rays. And then these high energy electrons will either directly or indirectly cause double strand breaks in DNA, and that's what will lead to the cell death. But it's important to keep in mind that there's lots and lots of factors that affect how much cell death actually occurs with the same amount of radiation. So we've seen cell cycle, oxygenation, and the linear energy transfer, and these can cause many factors difference in how much cell death you get. Okay, so just check the time. Oh, cool. Okay, so the second part that we were going to go through is how do radiobiology models improve radiotherapy treatments? So, with this information and with those surviving fraction curves that you saw in the first section, how do you actually use that information to affect treatment? So I'm going to introduce you to new type of graph now. There's a lot of graphs in radiobiology. And this graph shows tumor control probability and normal tissue damage probability. So how you read this curve here is, like before, on the x-axis we have dose, and the blue curve here is called the tumor control probability curve. So this tells me what percentage chance I have of killing a tumor given the amount of radiation. So if I have a tumor, if I give it 1,000 gray or infinite amount of gray, my chance of killing the tumor, along with probably the person, is going to be 100% if I give infinite amount of radiation. And if I give zero amount of radiation, the chance that I'll kill the tumor is 0. But of course, we're interested in this region here, this transition region, where some amount of radiation is going to produce some percentage chance of killing the tumor. The second curve here is this red curve, which is the normal tissue control probability. So in a similar vein, if you have a healthy lung and I give it, 1,000,000 gray, a ludicrous amount of radiation, the chance that I'm going to damage that tissue is 100%. And if I give 0 gray, the chance that I'll damage the tissue is 0%. And again, we're sort of interested in this build-up region where in between. So the idea, and that's this dotted line, is that for each tumor type that we're trying to cure, there is some amount of radiation that we can give that will have a high percentage chance of controlling the tumor, and just as importantly, a low percentage chance that will cause damage to normal tissue. So really, all the advancements in radiotherapy that I'm sure you've learned about in all the previous lectures and everything that we're doing at ImageX, everything that we strive for in radiotherapy is really to try to move this blue line as far to the left as we can and move this right line as far to the right as we can. And as we do that, this kind of area where we can successfully treat gets higher and higher. And that's sort of what we're trying to do. And what radiobiology does is it tries to model these curves in a way that you can predict what happens. Okay, so the fundamental mathematical model for radiotherapy and the fundamental mathematical model in radiobiology is called the linear quadratic model. So it's kind of very simple mathematics, and yeah, it's quite amazing that such simple mathematics can have such a profound effect on healthcare, really. So if you think of cell survival in terms of strand breaks, what you can do is graph cell survival in terms of strand breaks and in terms of dose in 2 components. So if we have a certain amount of dose, which will cause a certain amount of cell survival, which is this curve here that we've seen before, this curve we can really think of as having 2 components, the linear component and the quadratic component, which is why it's called the linear quadratic model. So the linear component represents when a DNA undergoes single lethal hit. So that means a single electron will interact with DNA, can be direct or indirect ionizations, as we've seen before. And that electron by itself will cause a double strand break. And this is linear in that if I have more electrons, I'm going to have more of these events occurring, whether they're common or rare. It's just a linear relationship. How susceptible a certain tissue is to this effect or is determined by this coefficient alpha. Okay, so it's alpha times dose. The second component is quadratic component, which represents kind of interactions that build up over time. So rather than a single electron causing a double strand break, this represents electrons that can interact just once and cause a single strand break. But then later, and it could be seconds later, it could be hours later, it could be days later, a second electron will come and then cause the second strand cause the second strand break. So this is quadratic because this is something that sort of builds up over time. Okay, so basically these combine this linear quadratic model and its two coefficients, alpha and beta, can describe more or less the surviving fraction of all different cell types. So people in radiobiology actually do this, measure this empirically. So what they do is they plate cells. They'll get tumor cells or even healthy tissue cells. They'll plate them on a dish and grow them. And then they will irradiate them. And then they will count how many cells survive. So that's usually in terms of colonies. So you would say a cell culture that has received no radiation after a week will have this number of colonies. But if they receive 4 gray or 6 gray or 8 gray, they'll have this many colonies left. And by doing this, which is a lot of work, we've basically built up a library of these alpha and beta ratios for a bunch of different cell types. And when you're doing radiotherapy, you use these kind of alpha beta values to basically try to predict how a person will respond if you were to irradiate them to try to cure their disease. Okay, so the general rule is that because the beta value is an accumulation, so it's based on time, tissue or tumor types that are able to repair themselves will have high betas. So this means that their alpha to beta ratio will be low. So late responding tissues, healthy tissues, these will have steep curves because the quadratic component is very high. So if you kind of imagine this blue line as being built up of a linear component and a quadratic component, a high beta means that it will start off kind of slowly, but then it'll accelerate really quickly as the quadratic component kind of takes over. A tumor, however, is not as good at repair as healthy tissue. So this means that in terms of alpha beta ratio, the alpha value is going to be higher. So in terms of this surviving fraction curve, it's going to stay flatter for longer. Of course, eventually the quadratic component will take over because it just scales so much higher. But this kind of initial flat section stays a lot longer because the alpha value is higher. Okay, so if you are following along, and I hope you are, you might see that we have a problem here. So the goal of radiotherapy is to kill the tumor and not cause damage to healthy tissue. But if you look at this linear quadratic model, if I give a certain amount of dose, for example, where this black line is, I'm going to have very low surviving fraction on this blue curve, which is the healthy tissue, and I'm going to have high surviving fraction on this orange curve, which is my tumor. So really, what we're doing is having the opposite effect of what we want. If I irradiate a volume, I'm going to kill a lot of the healthy tissue and not kill much of the tumor. So this is where the solution comes in, which is fractionation. So fractionation is kind of fundamental in radiotherapy. When a person gets radiotherapy, they usually get their radiation in lots of small parts. And that's to get this fractionation effect. So if you look at this blue curve, which is the healthy tissue, it accelerates very quick. But what fractionation does is you stop, rather than giving the amount of dose you want to give in one go, you stop after a certain amount of radiation. And then you have a break, and then you give your second fraction. And what this does is, rather than kind of continuing down this curve, you start this kind of linear quadratic curve all over again. And this kind of flat shoulder repeats itself. So in a radiotherapy treatment, rather than whacking all the radiation in one go, you get a bit of radiation and you have a break, and you continue and you continue and continue. And what this does is you constantly get this shoulder effect. Every time you receive radiation, you get this shoulder effect over and over again. This is for the healthy tissue, this blue curve, but the fractionation also happens for the tumor curve, this orange curve. But the key here really is that because of the linear quadratic model, this flat shoulder is a lot shallower, sorry, it's a lot steeper in this tumor section than it is for the healthy tissue. So if you look at this chart here, which shows fractionation really well, what we can do is use this shoulder effect to get rid of that problem. This problem that we had before. By using the shoulder effect, we can actually push this blue line above the orange line by taking advantage of this shoulder. So you can see here, if you look at the fractionated lines here, because of this shoulder, this purple line, which is normal tissue, has been pushed above, in terms of surviving fraction, the tumor cells. Whereas before, down here, when we have all our dose in a single go, the healthy tissue is still below the tumor cells. So by doing this fractionation, we've managed to move this up. Okay, so fractionation. So just in summary, fractionation loses effectiveness of radiation for all cell types, but more so for fast responding types. So that's kind of taking advantage of this shoulder effect. The alpha beta values graph the surviving fractions. So based on this linear quadratic model, if you know the alpha and beta values of your healthy tissue and of your tumor, basically you can optimize this fractionation for the best treatment. Yeah, okay. So why does fractionation even work? Yeah, it's a fundamental part of radiotherapy, but why? So in terms of radiobiology, there are these 4 effects that explain why fractionation is effective, and they're called the 4 R's of radiobiology, which is something that's sure to come up in any radiobiology exam. So fractionation basically gives time for healthy tissue to undergo 4 different processes that tumor cells don't do as well. And with these 4 processes, that's sort of the manifestation of that shoulder that you see mathematically. Okay, so the first is repair. So repair is kind of the most easy to understand. You have DNA that has a lot of single strand breaks. In healthy tissue, a lot of those, if you give the tissue time, a lot of those single strand breaks are going to be able to repair. Whereas in the tumor, that is less effective. So as you get more repair, what happens is that double strand breaks are going to be less common. So cells will survive more if you give them time. So that's sort of the real world explanation of what you see when this shoulder happens. So every time you have a break, a lot of the healthy tissues repair, so that when you get another dose of radiation, a lot of that radiation that would have caused double strand breaks are now sort of only causing single strand breaks. Okay, so the second R, sorry, yeah. Okay, so the second R is called reoxygenation. So this goes back to what we covered in the first part, which is that oxygen is really important for cell kill. As part of the interaction between electrons in DNA, you need oxygen to achieve those strand breaks. So what often happens in a tumor is that it's not uniform. Rather than having sort of this clump of cells where every cell is the same, you have a clump of cells where the outside has access to blood and therefore oxygen, but the kind of inner cluster of the tumor is hypoxic. So it doesn't have access to the circulatory system and it's starved of oxygen. So what happens when you irradiate is that these oxic cells on the outside are going to undergo cell death because they are able to get those DNA strand breaks. If you were to dump all this radiation in at once, these hypoxic cells, they're not going to be affected. And this isn't, this is something that we see in real life. So there's been studies where hypoxic cells have been irradiated with crazy amounts of radiation, 80 gray, 90 gray, and they still survive because, the sort of DNA strand break interactions just can't happen. So what happens with reoxygenation is that these tumor cells, if you have a break, it allows these oxic cells to die. And then these previous hypoxic cells are able to reoxygenate. So they get in contact with oxygenated blood again, and they become oxic cells. And then when the second batch of radiation comes, you're able to repeat this process. And this way you're able to kill sort of the inner hypoxic cells in the tumor that you weren't able to do before. Okay, so redistribution. So this goes back to the cell cycle that we were talking about earlier. So if you think of a bunch of cells, for example, in a tumor, they have cells in every part of this cell cycle. So you'll have, it's random. You'll have, lots of cells in growth, a lot of cells in synthesis, and lots of cells in mitosis. So what happens is that if you were to irradiate all the cells at once, certain cells in certain cell cycles you just won't kill because they're more radio resistant. So you won't, for example, in here, cells that are sort of at the end of this mitotic phase, they're going to survive with greater percentage than the other cells. But if you have a break in treatment, what happens is that these cells are more likely to spread out again in this cell cycle, and you're more likely to kill all the cells again because you don't have a bunch of cells kind of stuck in one cell phase. And another thing that you can do is kind of more from a research point of view is try to kill all the cells in a certain phase. And then when you have the remaining cells in the certain phase, try to predict when those remaining cells are going to be most radiosensitive, and then irradiate them at that time. What you can do is try to time it so that you can irradiate at a time where healthy tissue is at its most radio resistant and tumor cells are at their most radiosensitive. Very difficult to do, but interesting area of research. Okay, and then the fourth one is repopulation. So again, this is a pretty simple one. If you give cells time, they're going to have time to regrow. And cells that are able to repopulate, you just need to know the way in which they repopulate. And then you can factor that into your treatment plan. So that the amount of extra cells you have because you're spreading out your treatment is sort of like a known quantity, so you can factor that in. Okay, so that covers fractionation. And then one sort of extra thing that you should know about is something called hyperfractionation. So hyperfractionation is basically the opposite of what I've just told you. So you know from the linear quadratic model that it's advantageous to spread your radiation out over a long period of time to try to take advantage of that shoulder. Hyperfractionation is sort of the opposite. What it's trying to do is to basically ignore the linear quadratic model. And this, I've put this in because it kind of represents where radiotherapy is going. So when radiotherapy started and when the linear quadratic model was what people were using to do radiotherapy, this is what treatment plans look like on the left. So if you have a certain area you're trying to irradiate, I'm not sure if you can see on Zoom, but it's these kind of two shapes here. You kind of just give this block. And then what you can see here is that tons of normal tissue is going to be irradiated. Lots of healthy organs are going to get damaged. What you're relying on is that linear quadratic model shoulder to do maximum damage to the tumor and try to minimize damage to healthy tissue. But really, if you look at any what radiotherapy is like now, and the plan is more like this. And we call this higher conformality. I know this is called conformal, but it's not really conformal. So in like modern radiotherapy techniques, we're able to shape the radiation beam with very, very high precision, millimeter precision. And what this means is that we're not really irradiating much healthy tissue anyway. Most of the dose where deposit is able to be really highly targeted geometrically to the area we want. And this means that the linear quadratic model, sort of that shoulder is a little bit less effective. So hyperfractionation is very popular nowadays because it allows patients to get their treatment done, like over and done with quickly. So rather than relying on the linear quadratic model, instead of delivering kind of these big fields, you're trying to deliver small radiation fields. Some can be as small as like 3-4 millimeters, or to deliver treatments that are very tightly conformed, so exactly to the shape of the tumor. And then because of this, you can sort of ignore like that shoulder effect and do your treatment in 3 to 5 fractions rather than kind of 30 fractions, which was the case before. So because this is sort of new area, this also changes how the body is going to react. So in terms of radiobiology research, a lot of the research now is about how to model radiotherapy in its current form, where the dose is delivered a lot faster in a lot more kind of geometrically targeted technique. Okay, so that covers the second half of the lecture, and hopefully you can answer the second part of radiobiology, which is how do radiobiology models improve radiotherapy treatment? So the linear quadratic model is what mathematically describes how cells will respond to radiation. And fractionation, which is that shoulder effect, it's the shoulder effect mathematically, but in real life, it allows the cells to do the 4 R's, which is repair, reoxygenate, redistribute, and repopulate. And then based on the knowledge about alpha and beta values, you can optimally fractionate a tumor. So you can maximize the tumor control probability and minimize the normal tissue complication probability. And that's how you sort of get the optimal radiotherapy treatments. So when you hear that we're going to treat this tumor with certain amount of radiation, say we'll give this tumor 30 gray. This isn't something that's just been plucked out of thin air. These numbers have been worked out through kind of experiment and through mathematical modeling. So thank you for listening, and if you have any questions.
Thanks, Paul. So I'll open now for any question in the chat. All right, and also while I let you think about it, I'm gonna put Julia's email in the chat for those who want the attendance certificate. So one thing that we haven't really touched about in the summer lecture series is FLASH. So for those who don't know, FLASH is a very, very high dose rate irradiation. I was just wondering if you can talk a little bit about the radiobiology of that and how FLASH might be better.
Yeah, I think the thing with radiobiology with FLASH, well, so for those who don't know, FLASH is about delivering dose extremely quickly. So, you know, rather than sort of one gray a minute, we're talking, you know, 1 gray, you know, tens of grays, or maybe even hundreds of grays per second, and just sort of getting the treatment done immediately. The thing with radiobiology about FLASH is that there is no radiobiology knowledge about FLASH. Yeah, so FLASH is one of those situations where if you go by the old radiobiology, much like hyperfractionation, you expect, you know, poor results. Yeah, if you go by the linear quadratic model or models that people have used, you know, you expect basically worse outcomes or than what you get. But sort of in the early experiments of FLASH, where they've done animals, you've got kind of excellent results. And yeah, I think going back to what I said, so at the very, very start, radiobiology research is really hard. It's really slow. It's a lot of plating cells, irradiating cells, waiting, and then getting a graph with like 2 points on it, and trying to get results. And yeah, so I think in terms of FLASH, it's big, you know, it's a in terms of radiobiology, it's a question mark as to why we're getting these positive results. And there's a lot of work, you know, to be done to try to figure that out. Cool,
awesome. Does anybody else have a question for Paul?
Yeah, Paul, so I maybe missed that point. So what happens to the shoulder effect with hypofractionated treatments?
Yeah, so the shoulder effect, if I just go back. So the shoulder effect still happens. But the idea is that with these new treatments, the healthy tissue is getting so little dose that it almost doesn't matter. So if you look at the left, the healthy tissue, so anything that's not in this kind of U shape, it's getting irradiated with the same amount of radiation, but you're relying on that shoulder effect to try to kind of not have normal tissue damage. With hyperfractionation, you're basically letting that damage, you're letting that shoulder effect not happen, but you're replacing it with conformality. So you're shaping the radiation so closely to your target volume that the normal tissue isn't getting radiation anyway, or the radiation is getting is really, really low, that it kind of mitigates the linear quadratic model in that way. So I guess it's kind of using geometry rather than using time to kind of get around that problem.
Oh yeah, yeah. Thanks.
All right. It's 3:56. So unless somebody has a quick question for Paul, then thank you everyone for attending. Oh yeah.
I can ask a very kind of stupid question, probably. So like when you do these experiments for radiobiology, you probably do this for like you just use some animals to do these experiments, right? And then how does that translate to humans? And
So yeah, so one of the things with radiobiology actually is that the experiments aren't done in animals. They're done in petri dishes. So those alpha and beta values, and that's kind of one of the criticisms of the linear quadratic model, is that you've taken the cells out of the environment that they're in, that you're trying to model. So when you do these experiments, you take these cells and you plate them on petri dishes so you can assay them and count them and see how they survive. So this means that you can get very accurate cell counts. But yeah, it's not sort of in the environment of an organism anymore.
Yeah. Okay. Cool. Thanks.
All right. Thank you, Paul, for all the information. Again, I've put Julia's email in the chat. If you want an attendance certificate, please send her an email. I will leave the Zoom running for another minute or so, just so you can note the email, but otherwise feel free to leave.
Thanks, everyone.
Thanks, Paul.
See ya.
See ya. Alrighty, I'm gonna end the Zoom meeting now. So thank you, everyone, and see you maybe next year.